Hi, the next topic I want to cover are the major viral characteristics, and so these are the things we might use to describe a particular virus. And in addition, after that, we'll get into phylogeny of viruses and taxonomy of viruses. And we're just really going to scratch the surface of those two topics again. We're not going to do any detailed analysis. We're going to learn some terminology and some very important basic facts. So let us begin. The first characteristic to look at is tissue tropism or viral tropism, and this talks about which types of cells a virus can infect. So in particular about an animal virus, so a virus that can infect animal cells like human cells, there are a lot of different cells to choose from, and which cell a virus is able to infect really determines what kind of immune response we get, and it determines what signs and symptoms get caused. For example, the viruses that cause the common cold, of which there are hundreds, they all affect upper respiratory tract epithelial cells. And because of that, they all cause the same immune response, and so we all get the same disease and call it the common cold. Contrast that with a virus that infects, cells in the digestive tract, in the stomach, and in the small intestine, norovirus. That's going to cause vomiting and diarrhea. So a lot of that is just caused by which cells the viruses infect, and that is their tissue tropism, or their tropism. And on this slide, I mention some more details about it and some things you can use to predict a tissue tropism. Like, for example, once we found out that SARS-CoV-2 virus, which causes COVID-19, once we found out it uses the ACE2 enzyme as part of viral entry, and it needs the receptor TMPRSS2, once we know that, we can figure out what cells in the body have those two things on their surfaces, because that's going to tell us which cells it can infect. And for SARS-CoV-2, it makes perfect sense because there are those proteins are on the surface of cells in all the different tissues that virus infects. And different strains of COVID-19 needed they might depend more on the ACE2 or more on the TMPRSS2, and different cells have more of one than the other, and so the different strains have different effects mostly because it affects different cells. And similarly, influenza viruses are going to have very specific molecules they attach to, and that really limits what kinds of cells they can get into. So again, the way we think about it when we're thinking about a disease is like, what kind of tissues do they infect? And the way we think about it when we think about viral replication and disease transmission and things like that, then we think about what surface proteins do they need for getting into a host cell. We can think of it at those two really different levels, but either way, it's tissue tropism. A more broad way to describe a virus is its host range. So what kind of host organisms does it infect? Every virus that infects humans has humans as part of their host range. So the host range doesn't tell you what part of the body it affects, it just tells you what species. And this can be really different from virus to virus. So we're going to look at some bacteriophage. Those are viruses that infect bacterial cells. So none of those are going to infect us. There are viruses that only infect members of the archaea, or fungi and protists, or plants or animals. Most individual viruses have a host range way more narrow than one of these groups. There aren't many viruses that are going to infect all bacteria, or all fungi, or all animals. And we can never really predict the host range of a virus. So on one end, we'll have really broad range viruses like the rabies virus, which can infect most mammals. It's a big group. And that matters to us because we can get the rabies virus from bats or from dogs, or from raccoons, or from other people in really hideous circumstances. Influenza can infect birds and pigs and humans, and that means we keep getting influenza. We keep getting over it as humans and then getting it back from pigs and birds. So that broad spectrum really does matter in terms of our relationship to a virus. Other viruses have incredibly narrow range. Like the bacteriophage lambda is one of the best studied bacteriophage, and it only infects certain strains of Escherichia coli. So that's certain strains of one bacterium, one species of bacteria. So everywhere in between, there are viruses with different host range. Another characteristic that we'll always use to describe a virus is its genome type. So you'll remember I mentioned that a capsid will have the genome inside it, and the genome could be DNA or RNA. Now that's kind of weird, right? There's no bacterium that has an RNA genome. There's no human cell that has RNA instead of DNA, but some virions have RNA instead of DNA. And that does determine how they have to replicate inside a host cell. It really affects what they have to do in a host cell. So again, RNA or DNA, those are the choices, but also it could be one or two strands of both of those. It could be double-stranded DNA or dsDNA, single-stranded DNA, ssDNA, double-stranded RNA, dsRNA, or single-stranded RNA, ssRNA. So that's how we abbreviate them, and we can come up with examples of all of those. Then there are also viruses we think of differently, like retroviruses, because they have RNA genomes in the virion, but then they do reverse transcription. As soon as they get into a host cell, they make a DNA copy of it, which never occurred to most people until it was discovered. And that's actually a clear example of some nerd going out to try to find something weird, doing research. And if that person hadn't had that idea, we would have no idea what HIV is. We'd have no treatments for HIV if some nerd hadn't gone out wondering, are there viruses that do reverse transcription? That term didn't even exist. That's me on my soapbox. Anyway, within the RNA genome viruses, there are really two very different types. And this is where you'll see plus sense or minus sense single-stranded RNA. And this really dictates a lot of, again, a lot of their behaviors inside a host cell. So the difference between plus sense or minus sense is whether that RNA can be translated directly or not. And inside a normal cell, in bacterial cell or human cell, normally every RNA that gets made can be translated. They're all plus sense RNA. They're messenger RNA. It's all plus sense. So we don't think about this unless we're talking about viruses. But some virions have minus sense single-stranded RNA. That means when you put that RNA inside a cell, ribosomes can't translate it. They can't do anything with it. And first, before they could translate it, a viral enzyme has to make the opposite strand, and then a ribosome can translate that. So in the next video, which is really optional, I go into biosynthesis in detail, in a lot of detail, way more than I typically want students to go into for this class. Once you go through that, you can see really the difference between a plus sense RNA and minus sense RNA and why it matters for biosynthesis. For our point of view, the simplest explanation you need is that positive sense RNA is RNA that can be translated directly as it is, and minus sense RNA cannot be translated as it is. The virus must somehow make a copy of the complementary strand once it's inside the host cell. So those are the genome types. Another really important characteristic of some viruses is viral latency. That is, they can hide in a host cell for a long time. Maybe in the case of bacteria, they can hide in a host cell for many generations. In a human, some of them can hide in a host cell our entire lifetime. And during that time, they're not making virions. They're not going through the replication cycle. They're doing something else and waiting. So many of them will actually copy their DNA in a way that puts it into the host cell's DNA genome. So the host cell chromosome now has the virus DNA in it, and a lot of human DNA is actually virus DNA, ancient virus DNA. And so I cannot stress this enough. When a virus incorporates its DNA into the host DNA, there's no more virion. There's no other physical form of that virus, but the virus is still a virus. At any moment, it could be translated, and virions could get made by that cell, and the virions could escape and go infect other cells. But for a long time, that doesn't happen. I get really excited when I think about this, because this is where you see that a virus is just information. A virus can exist just as a nucleic acid sequence that we could describe as a sequence of letters. It could exist in a host cell for many generations of that host cell. There's nothing else on Earth that can do that. There are no cells that can exist without a cell. Even an endospore still has a membrane, and it still has a lot of proteins, and it still has the ability to, under its own power, transcribe and translate when the time is right. And it is always its own physical form. An endospore is its own physical thing. But a virus can exist just as DNA inside a host cell. That is amazing to me. So again, there's no virion. And some examples of this would include HIV. That's a good example, that as soon as it gets into a cell, it puts a copy of its genome in the host's DNA. Other forms of latency exist where the cell doesn't put its DNA into the host chromosome, but instead somehow protects its nucleic acids long-term inside a host cell. So one example from the varicella-zoster virus, which causes chickenpox and shingles, this virus will keep a neuron alive, so it'll prevent the neuron from doing apoptosis, and it can keep its DNA inside the neuron's nucleus. How that happens is complicated, but it's able to keep that DNA in the neuron's nucleus, keep the neuron from dying, and that means that decades later, it can reactivate, start making virions, and a person will experience shingles. So those were the viral characteristics, how we would describe viruses. This is virus phylogeny, and there's a lot to it. You can study the evolutionary relationships between viruses, because viruses definitely evolve. But viruses don't all share a common ancestor. They don't all come from one original virus. There's no evidence that they do. And none of them share a common ancestor with any cell. So they are not like cousins of cells. They are pieces made from cells. So they're structures made by cells, but they're not like cells or related to cells. So nowhere on this universal phylogeny will you find a virus, but you will on this universal phylogeny find every cell on Earth, but no viruses. And so I mentioned you can study evolutionary relationships between viruses, and here is an example phylogenetic tree of negative sense RNA viruses. And it shows different virus families and how related they are to each other. So we have on the one side, we have The segmented genome ones seem to be closer related to other segmented genome ones, including Orthomyxoviridae, the family the influenza viruses are in, and arenaviruses, where the Lassa fever viruses, it's like Ebola. And then on the other side, non-segmented viruses, various other virus families, including the family that has the Ebola virus, that's horrifying, and the family that has the rabies virus, which is incredibly horrifying. The rhabdovirus family that rabies is in has examples of viruses that infect plants or protists. They don't all infect just humans. There's a lot of other viruses besides the ones we know and the ones that affect humans. So you may well have two closely related viruses with completely different host range. So that's virus phylogeny. I mostly want you to remember that there's no place for them on the universal phylogeny, but they are related to other viruses. It's just that you won't find a connection between this phylogeny of the negative sense single-strand viruses. You won't find a connection between these and the plus sense RNA viruses or the DNA viruses. They don't seem to be related. And then viral taxonomy is a necessary field. We have to be able to name viruses and keep them straight and communicate about them. There are so many viruses, and they evolve so much faster than cellular life does. The taxonomy is different. Most levels you would italicize virus groups, which we don't do for things like bacteria and animals. And virus species get descriptive names like severe, acute, respiratory syndrome, virus 2, SARS-CoV-2. And within those you get, viral variants, viral strains, and it's just kind of crazy because within one species or subspecies, SARS-CoV-2, there are thousands, maybe millions of strains of that virus that we never usually hear about because many of them are redundant. So that's all I want you to know about viral taxonomy, and this video has hopefully given you a little more insight into viruses and how we describe them and how they're related to each other. In the next video, we will look at virus replication, and that's where we start to really see how weird they are. So I'll see you at the next video.